Welcome back to the Neurostimulation Podcast. I'm Dr. Michael Passmore, Clinical Associate Professor in the Department of Psychiatry at the University of British Columbia. Today's episode is going to be a fascinating deep dive into one of the most exciting and rapidly evolving areas in neuromodulation, transcutaneous vagus nerve stimulation, or tVNS. My guest today is Dr. Ronan Denyer, who recently completed his PhD in neuroscience at the University of British Columbia under the supervision of Dr. Lara Boyd. His work integrates brain stimulation, neuroimaging, and behavioral neuroscience to understand how the brain prepares and executes movement, particularly through the dorsal premotor cortex. He's now a postdoctoral fellow in the Coactions Lab working with Dr. Julie Duque, where his research is expanding into the intersection of TMS, vagus nerve stimulation, and the locus coeruleus noradrenaline system, a key neuromodulatory system involved in things like attention, arousal, and decision-making. And so one of the papers that we're going to be discussing today explores how non-invasive vagus nerve stimulation can actually improve perceptual decision-making accuracy, shedding light on a long-standing debate about how the locus coeruleus shapes human cognition. Ronan, welcome to the podcast.
Yeah, thanks so much for having me. Looking forward to the conversation.
So maybe if you can just provide a bit of an explanation about your academic background and some of the things that your lab is currently working on, just so that we can get to know you.
Yeah, sure. So I'm from Ireland. So I grew up in the northwest of Ireland and actually pursued my undergrad in Trinity College Dublin. And so during my time there, in my final year, had to do a final year project. And so I ended up working with a professor there, Richard Carson, and he works a lot with transcranial magnetic stimulation within the motor control space. And so that's where I started my journey with brain stimulation, and I enjoyed the process. And then through Richard, I ended up being introduced to Lara in UBC. And I decided I wanted to pursue a PhD, and it just ended up working out that I ended up in Vancouver. It's a nice coincidence. And so, yeah, in Vancouver, I worked a lot with TMS. My thesis work was more focused on kind of young, healthy populations, on more basic neuroscience questions, motor control questions, bimanual coordination and stuff like that. And then I also, working in Lara's lab, worked quite a bit with clinical populations, particularly people who've had a stroke. So trying to apply the methods in that population too. And then, yeah, after I finished my PhD, I decided I wanted to move back a bit closer to home. So I was looking through potential locations and ended up in Brussels with Julie Duque. And yeah, just when I arrived, a former postdoc who has just moved on to another position, Sue Xiang, she had just finished a project looking at using tVNS within a decision-making paradigm. And I wasn't exactly sure what I was going to do when I joined the lab, because I had just finished and fully figured out what experiments I wanted to do. But I was very intrigued by her results using tVNS, and then I thought about pursuing that. So yeah, that's how I ended up working in tVNS. It wasn't like this sort of grand plan. It was, they're doing it here, and it seems like an interesting area. So yeah, that's how I ended up working with the method.
Yeah, cool. Thanks for explaining that. It is really interesting how an academic journey can go in that way. You make connections, you network, and then you get drawn in certain directions. And so I'm excited to discuss current projects. But one of the papers in particular, just for viewers and listeners, as a kind of a preview, is that I think I'd like to explore today is this idea about how non-invasive vagus nerve stimulation can actually improve perceptual decision-making accuracy, which is so interesting. And I think it helps to shed light on what I understand is a long-standing debate about how specific areas of the brain, like the locus coeruleus, can shape human cognition in general. Yeah, so maybe why don't we just follow up with that? So for viewers and listeners who are not as familiar, and I don't admit to being particularly familiar with these specifics either, so why is the locus coeruleus specifically such a big deal for that, what we're talking about in neuroscience in general?
Yeah, so it's a really interesting part of the brain. I think the reason why people are interested in is because it has this reputation as a very small nucleus, but very big influence. So it's really just in the brainstem, posterior part of the brainstem, and it's very small, and it has this blue color, which is actually what it means in Latin. Locus coeruleus means blue place. And so if you were to do dissection, it's blue because actually there's a lot of neuromelanin in that area. And the reason there's neuromelanin is because the cells there are noradrenergic. So it's a byproduct of the process of creating noradrenaline is to have this blue color. And so that's really what those neurons do. They reach far and wide into the central nervous system, from the spine into subcortical regions, and then also to the cortex. And so they have the capacity to modulate activity in almost every part of the brain. And so that's how it has this potential for a big influence. And then on the other side, it receives so many inputs from lots of different regions too. So it's just like this very dynamic region. And so, yeah, it's interesting because I would say, it has this big influence, but the specific function isn't that well understood. And so that's opened up a lot of different questions. And then I think there's also this other kind of clinical angle that people are interested in it, because when we're thinking about brain stimulation in clinical populations, often researchers are looking for regions that we can stimulate to maybe induce some sort of increases in activity or to induce plasticity. So I think that's another reason why people are interested in the region.
Yeah, for sure. Thanks for explaining that. And yeah, it does remind me a little bit of some forces in that the locus coeruleus, as you say, primary source of noradrenaline or norepinephrine. I can never remember, is noradrenaline the European phrase and norepinephrine the North American phrase?
Yeah, I think that's the way it is. Yeah, it's the same thing, but yeah.
Right. So it's interesting because, yeah, as you say, it's such a kind of a way station in that sense, an important way station in terms of the global modulation of brain function, but particularly for aspects of, I suppose, mind and consciousness that relate specifically to the noradrenergic system, attention, motivation, mood. Yeah. So walk us through a little bit about tVNS then. So transcranial vagus nerve stimulation. Yeah, what exactly is it, and how does stimulating the ear actually influence the brain? Because it intuitively may not exactly make sense. So help us to understand how that works.
Yeah, so I think, I'm not sure what the order was, but there's also an invasive version of vagus nerve stimulation where you actually do a surgery and implant a stimulator onto the vagus nerve. Similar to, I'm trying to remember what you call the thing you implant for a heart pacemaker. Yeah, similar kind of surgery. And so the idea is that you stimulate the vagus nerve. The vagus nerve actually has connections into an area called the nucleus tractus solitarius in the brainstem, and that itself connects to the locus coeruleus. It's a way to increase activity in the locus coeruleus, but of course, it's a very invasive procedure. It's more so like a kind of experimental clinical procedure for now. We can't just have people come into the lab and do that type of experiment. So there's been this idea of, is there other ways that we could stimulate the vagus nerve? And so the vagus nerve actually has connections with the ear, so afferents from the ear into the vagus nerve. And so in particular, the kind of central part of your ear here, called the cinna conchae, and around the tragus part of your ear, has these nerve endings which connect with the vagus nerve, a branch of the vagus nerve. And so the idea is that if we stimulate those, can we stimulate up into the nucleus tractus solitarius and then also the locus coeruleus, and provide a way to modulate activity and increase release of noradrenaline in the brain? And so that's where it all started. And yeah, I could keep going, but that's where we started with tVNS and why we were looking at the ear.
Yeah, thanks so much for explaining that. I think that's extremely helpful to differentiate from implanted VNS as well. It makes me wonder, there's this newly approved device called the ProLive RX. I'll have to just refresh my memory about that, but it strikes me that perhaps that's a similar, this is for depression. And I just think that there's probably a similar kind of approach as far as the non-invasive cranial nerve stimulation to improve mood. But yeah, maybe I'll clarify that, and maybe I can try and link to that if there's relevance at some point. But then just getting back to your lab's research, maybe, what would be a way, I guess obviously there's a difference between the specifics that your lab's been looking at and more broader clinical applications, although obviously as you're describing. That's part of what the research is geared towards furthering, is just the knowledge translation there. But how would you say that the transcutaneal VNS could be considered in comparison to other technologies like transcranial magnetic stimulation or transcranial direct current stimulation, those kinds of techniques that are being more routinely applied in a clinical setting?
Yeah, so on the face of it, it's the kind of physics of it are quite similar to TDCS, I would say, and where you have an electrode on the skin and another one near to it on another part of the ear, and you're just passing current through, which is increasing activity in the vagus nerve, and then up into the locus coeruleus. I suppose with TDCS, it's more like you're just directly hitting the cortex. You're not doing this kind of other pathway into the subcortical region. And then TMS or repetitive TMS protocols are more similar, but it's a magnetic pulse and it's a pulsing. It's not like a continuous, you're creating an electrical field using a magnetic field. But yeah, I think it's probably more similar to TDCS, but it's its own thing too. I think one thing I read that is appealing to me about the method is just being able to measure response, because the locus coeruleus has this very clear actual connection with our pupils. And so if you change activity in the locus coeruleus, there's these kind of like maybe 3 or 4 synapses away connectivity with the muscles, which increase and decrease the size of our pupils. So I think one real benefit for tVNS is that if you turn it on, you can really track the effect it's having in a specific subject, because you can just use a pupillometry setup to measure changes in the eye, or in the pupil rather. So that's something that really appealed to me, having used repetitive TMS in my PhD, been a more trying to do like a virtual lesion and see how it affected behavior on this particular task. It's often difficult or expensive to really understand what the stimulation is doing to the brain. And so you sometimes have to take a bit of a leap of faith, whereas I like the tVNS because you have this clear, you're measuring once every millisecond, getting this kind of clear picture of what's happening, at least just from looking at the pupils. So I think, yeah, that's maybe a sort of an edge that tVNS has, maybe.
Yeah, that's super interesting. Those little details, it's fascinating to understand the specifics of that. And I can completely understand your perspective on, yeah, the advantages there for sure. So one of the papers that I was interested in talking about with you is the publication in the journal Brain Stimulation called Transcutaneous Vagus Nerve Stimulation Boosts Accuracy During Perceptual Decision Making. And yeah, maybe if you're able to help us to understand the idea behind the particular study and the results and why that's an important paper.
Yeah, so I'll do my best. It was just after I got here, but I'm fairly familiar with the work. And so basically the idea is to look at the effect of tVNS on what we call perceptual decision making. And so what we mean by perceptual decision making is really what we call a random dot motion task. And so just for the listeners, the viewers, basically the participant is sitting down and they see this kind of cloud of dots basically in the center of the screen, and they're able to respond left or right. And so they just see the dots moving in these random directions at first, and then at some point they get a cue to and are told that the dots are going to start moving in one way or the other, and they have to do their best to estimate if they're moving left or they're moving right. And it's basically calibrated for it to be quite difficult, where a certain proportion are moving right, certain are moving left, and you have to do your best to assess basically which direction they're moving in. And it's a nice task because it's been used a lot in neuroscience. So there's all these kind of ways in which people model the data, established methods for modeling the data. But I would say basically what Sue found in her study is that what she did is basically did the task like that, but she had 2 conditions. So in one condition, just before they had to start deciding or make a decision, the tVNS was turned on for 4 seconds, sort of burst of 4 seconds on each trial. And then there's a kind of control condition, so just a sham stimulation condition. And we need to have a sham control because just having this sensation on your ear can maybe in of itself change your behavior without maybe having these direct effects on locus coeruleus that we think it might have. So we compare the results against a condition where we stimulate just on the earlobe. And so that part of your ear is not innervated by the vagus nerve. So it's a nice control because it's similar sensation, but it doesn't have this vagal aspect to it. And so when she looked at the data, what she found was people were significantly more accurate. Now, not by a huge amount, but were more accurate with the tVNS versus the sham. And sometimes when you see a change in accuracy in a task like this, it can be strategic where instead of they're more accurate, but they're also slowing down their responses. So they're allowing more time for the evidence to accumulate. But she didn't find any difference in the reaction time. So it's like they just got this free boost in the accuracy of their perceptual decision. And yeah, it's just, I guess there's evidence that the tVNS is boosting the decision process in some way. And then she went a bit more deep into the data and found that it was, the effect was even stronger when considering basically how people reacted after an error. So you have some people who, after they make an error, they become they're negatively affected by it, and it decreases their performance on the next trial. Then you also have some people who, if they get something correct, then they relax, and then that actually they see a decrease in their performance on the next trial. And so what she found is, like, by grouping people into those groups, the tVNS particularly had a strong effect when in these challenge modes where you typically see a drop off in sham. It had this kind of rescue effect, you could say. I think you have this kind of global effect on accuracy, but it might be driven more by these specific circumstances where there needs to be this input, or there can be this boosting effect. So that's, very interesting. It speaks to the more global view on what this neuroadrenaline is actually doing. There's this idea that the function is really it boosts ongoing activity, and so reduces the signal to noise ratio, or increases rather. And so that's how you get this free accuracy. So that was the crux of it. And so from there we've gone a few different directions, but that was the foundational work, and we're really getting into tVNS.
Yeah, that's fantastic. Yeah, thanks for explaining that. And it'll be great to talk more about the other directions that your lab has gone in since then. But it's so interesting because I think, it was a great explanation of this historical controversy to a certain degree between the gain hypothesis, or the improvement in the signal to noise, which is leading to better accuracy, that the locus coeruleus and the neuradrenergic system is implicated in, versus this kind of more of an urgency hypothesis where decision making is accelerated but potentially less accurate. And as I was understanding, so this finding about the findings highlighted that there was, as you say, that improved accuracy but not really a change in reaction time, which I suppose supports the gain hypothesis.
I think so. You're not seeing a slowing or a quickening. It's just, in fact, that use this model called the drift diffusion model to model this data, and it assumes that there's some sort of decision process going on, and so the evidence is accumulating at a certain rate, and then eventually you hit these bounds which are represented in the brain somewhere, and that's what drives the decision. And so Sue did some modeling using that method and found that the drift rate increased. So the speed at which evidence is accumulating is increasing, and so that leads to more accurate decisions.
Yeah, and it was really interesting, the finding that the effect was strongest after errors, and it makes me wonder if maybe the tVNS is helping people to recover from attentional lapses, or that kind of thing, some subtle things that could be speculated.
I think so. I think the way I'm constantly rethinking how I think about the region, but I think certainly it seemed like there's this kind of resetting process or something where, you maybe get lost in thinking about the error that you just created under sham, and then a sham doesn't help that, whereas the tVNS maybe locks you back in to the task, into the relevant aspects of the task, instead of this kind of process of thinking about the error. Yeah.
Yeah, maybe a metaphor. I don't know if this is, obviously it's too simplistic, but maybe something that can help viewers and listeners and me wrap my head around this is
Yeah.
maybe the locus coeruleus can be conceived of in the sense that we're investigating, or we're talking about that you investigated, that the locus coeruleus is less like a gas pedal and more like a signal optimizer.
Yeah. Yeah, I think so. Yeah, like it's really boosting what's already going on in the brain. It's not really changing the dynamics. It's more just coming in. If there's activity going on, that's going to be increased. If there's less activity between these synapses, that's decreased. And so you just kind of get this sharper, more HD signal. And so that has benefits. And then I think that there's also this energy management aspect to it, because I think a natural reply is, why aren't we just like that all the time? Wouldn't it be better to just be HD all the time? But we don't actually need to be. So that's why we have this pulsing. It's more so in circumstances that we might need this input to push our speed accuracy trade-off, not to navigate it. And so, yeah, that sort of makes sense in an energy optimization viewpoint, which the brain is very much is optimized to not use too much energy when it doesn't need to, and all that. Yeah, interesting kind of question.
Yeah, definitely. Yeah, so you alluded to just now some other branches, some other pathways that your lab has chosen to follow based on the findings there. So yeah, maybe talk a bit about some of the projects that you have going on now that you're excited about.
Yeah, I just finished and published a project using tVNS. Actually, it was when I just first got here, and I wanted to get going on something that quickly that I could have going while I also started to develop something else. And yeah, because I had this experience using TMS, I decided to combine the two. And it's a very basic question, but actually there just wasn't a lot of information out there. So what I was interested in is what's the effect of these 4-second bursts of tVNS on our corticospinal excitability. So I guess for some background, as I said earlier, locus coeruleus has all these kind of vast, broad connections. And so through kind of these anatomical tracer studies, it's known that there is these connections with the motor cortex, or direct connections in the motor cortex. So I was interested in, if we stimulate tVNS, is there a change in the balance of excitability in the motor cortex? And so when we use single pulse TMS, we can just do pulses over the motor cortex, and it creates like a twitch in the muscle, which we call a motor evoked potential. And so that is just a measure of what's the state of the corticospinal tract, or the excitability within the tract. So basically what I did was sat people down, just did trains on the ear, sham and tVNS. And at these kind of set time points within the train, I just did elicited MEPs using TMS, and then measured the size of the twitch and average all the data. And so what we found was, so we measured just before as a baseline, and then very shortly after the train on the stimulation onset, and then at 1 second, 2 seconds, 3 seconds, and 4 seconds. And then also at 5 seconds, so 1 second after the stimulation had finished, and at 6 seconds. This is all across different trials. And so what we found was there was an increase. The tVNS did create an increase in the corticospinal excitability, but it was really specific to the time points at which the tVNS was actually turned on. So not so much after it was turned off at those 5 and 6 time points, but more so in the train, particularly in the kind of last section of the train. So if we turn on tVNS and then we measure at 3, we make MEPs at 3 and 4. Those are typically bigger than the ones at the start of the train. And so, yeah, it's just showing that there does seem to be, when we elicit a change in activity in locus coeruleus, there does seem to be this increase in excitability in the motor cortex as well. So yeah, we wanted to really do that as maybe a foundation into seeing how this changes in behavior and stuff like that. But the TMS experiments are quite time costly, because you have to do a lot of different trials, because the MEPs can be quite noisy. And then with tVNS itself, we also measured pupil size. And between each of the stimulations, we wanted to allow enough time for the pupil size to come back to baseline. Yeah, ended up been quite a long experiment, but I think, yeah, it's an interesting outcome and opens new avenues, I think, and is also, I think, interesting from a clinical perspective as well, particularly for stroke and stuff like that.
Yeah, I was going to ask a bit about possible clinical translation. One thing I'm just curious about is the relevance of the pupil size, because it's come up a couple of times here, and I just was thinking for my own curiosity, but also for viewers and listeners who might be curious about what's the relevance of measuring the pupil size.
Yeah. So I suppose, given that it's known that locus coeruleus has this relationship with pupil size, where pupil size is really driven by changes in activity in the region. And this is known quite well established from animal studies, but also in human studies, like in fMRI studies, for example. If you do tVNS, you see an increase in pupil, you typically also see an increase in the brain region through fMRI. And so it's an established way of tracking the effect, or tracking if there's a change in the state of locus coeruleus. And so the reason we included it in the study is because it gives us a more of a gold standard of whether there is an effect. And so we did also see this clear effect in the pupil size too. So that gives us a good feeling that we're not just seeing things, that there is this clear effect on something that we would expect there to be an effect. And then we also have this effect on the motor road potentials too. So that's, and the thing is, it's experimentally, it's very easy to implement. You just have to quickly calibrate it, and it's just continuously recording, and you just get the data. It's quite easy to work with. That's why we like it.
Yeah, that's interesting. It's like a real-time live biomarker, just as a confirmation of a proxy, in a sense of just all the changes lining up and coming together. And that's really interesting.
Yeah, it was interesting. I think that there's actually some interesting stuff that came from it. It's good to see the effect, the clear effect, and it was actually very similar shape to what we'd seen in our previous experiment. One thing that was interesting is, even though at the group level there was both these changes in the MEPs and in the pupils, they weren't actually correlated, and the timing was quite different too. So with the pupil, you see a peak after about 1.5 seconds, and then it decays. Whereas, as I said, with the MEPs, it's more like a ramp, a slower ramp through the onset of the tVNS. So, yeah, that's interesting as to why both, as I said, MEPs are quite noisy, so that could be contributing. But there's also this idea that the locus coeruleus might have these different sort of nodes. So it's not like one area that kind of reacts to everything. There might be little nodes, and maybe one node is more affected, is having a bigger effect on the pupil, and another node is having a bigger effect on the motor cortex. So that might be why we see a lack of correlation. But there's a lot more questions, but we'll need to do more studies to fully figure it out.
Yeah, maybe how the thalamus as a sort of like a way station is all divided into various different components itself.
Yeah, exactly. Yeah.
In terms of that potential for clinical translation, I was interested in your team's paper suggesting not suggesting, but just explicating how locus coeruleus dysfunction is implicated in disorders like ADHD, depression, neurodegenerative disorders. So I'm curious, particularly in your work pairing tVNS with TMS, what would be some of your thoughts about where you might foresee that tVNS could be applied clinically?
Yeah, I think there's a lot of different possibilities. I guess I'm coming more from a motor neuroscience perspective, I think, and then having worked a bit in stroke. I think a lot of the time in neurorehabilitation and stroke, people are looking for ways to potentially change the excitation balance in the brain through brain stimulation as a means to promote plasticity. Because obviously after you have a stroke, it usually affects the motor cortex to some degree. And so I think there's this kind of idea, if we can find ways to target changes in excitation in that region through simulation, then maybe we could pair that with therapy, like bimanual, unimanual therapy or something like that. And then that would promote these kind of plastic effects that would help the brain to recover. So yeah, I think having seen this effect that we saw, that there does seem to be this effect on motor cortex, I think it's it's grounds to maybe continue to pursue that. And I think as well, the kind of fact that we saw the effects, what we call online, so when we're actually applying the stimulation, not after, is important too, because it suggests that it should be these bursts that you pair with some sort of behavior, rather than sometimes what you can see happen in rehab is like, oh, we'll sit the person down and we'll do a priming brainstem for 20 minutes and then we'll do therapy. But I guess this speaks to more maybe doing the two together. Well, whilst you're stimulating on the ear, to also be doing some sort of motor task. Like that's maybe a strength of the method, is that it's actually amenable to that, because it's quite, you can be mobile. You don't have to be with TMS, you're stuck under the coil a bit. And so, yeah, I think it's an interesting area. I mean, there's also the invasive vagus nerve that we talked about, and that's a focus as well in that region or in that zone. And I think there's starting to be a consensus that the two should be paired together within that literature as well, I think. So yeah, and then that's just stroke. I think there's also Parkinson's disease. So we know that Parkinson's disease is really driven by the motor, at least the motor symptoms are driven by the death or the degradation of the dopaminergic neurons in the basal ganglia. But actually, before that happens, there's also a degradation of the noradrenergic in the locus coeruleus. And so there's maybe possibility for exploring the use of tVNS in that disease as well.
Yeah, it's really interesting. I think obviously the advantages in terms of the possible applications with something that's non-invasive, that perhaps the device would be portable, something that people could use, as you say, while they're engaged in other kinds of rehabilitative activities, or even outside of enclosed clinical settings, something that might be possibly home-based with remote supervision like TDCS has become. So
Yeah.
that by itself, the technology lends itself to exploration or along those lines, I can imagine.
Yeah. Yeah, and we have a custom setup, but I think that there's ways to make it cheaper. And yeah, so I'm interested to see what happens in that area. Maybe I'd like to work with it in the stroke populations as well, maybe someday, because yeah, I think it does have some potential.
I can remember an episode from a few months ago with Dr. Bernard Hommel and Dr. Colzado. They were talking a bit about in China, where they're researching now currently, that they have these sort of helmets, for lack of a better term, that sometimes some young kids are wearing, and they're having them, you see them around in the schoolyard, and it's intended to help with ADHD or something along those lines. So this is the kind of thing that I wonder, because with do you think there may be some future applications, for example, like in attention deficit disorders or other kinds of maybe neurodegenerative disorders?
Yeah, I think there is a hypothesis about ADHD. For me, I think it's interesting, but I would say there needs to be quite a bit more basic work to really be sure if it's worth pursuing on a wide scale clinically. But I think there is a clear potential for ADHD, at least partly being driven by deficits in the locus coeruleus. So I think the idea with ADHD is, the locus coeruleus, there's a interesting kind of aspect about the region. It has these kind of different firing modes. So just at rest or in daily life, it has kind of this, what we call the tonic firing rate. And so this is just when you're in daily life, whatever the baseline firing rate is called the tonic firing. And then when you're engaged in a task, there's this phasic effect where there's this kind of sudden increase. And so I think the idea with ADHD is that, and so to go back, sorry, so the tonic firing is thought to relate to your arousal level, and it's said to follow this inverted U shape in the typical way we would think. So when your tonic firing is low, you're in a sleepy state. It's after you just woke up, or as you're supposed to go to bed. It might be promoting you trying to go to bed. And then in the middle, when it's at a moderate level, that's called optimal task engaged zone, where you have that feeling of being really locked in to a task. And then when it gets high, it's in this, where you can't really focus or having difficulty focus. And sort of this hypothesis about the tonic that it has this kind of meta-cognitive role mediating between explore and exploit modes. So when we're in an environment where we need to engage in a task to maximize rewards from the task, we're put into that moderate zone. Because it's moderate, maximize the effect of the phasic inputs when we're doing a task. And then as it starts to drift forward, maybe as the rewards are becoming more hard to come by, it's a signal to explore or to move somewhere else in our environment. I think the idea with ADHD is that, and I think there is evidence that the tonic firing is higher than a typically developing population. And so the idea is that because the tonic firing is higher, they're stuck in this explore mode. And so that's partly why they find it difficult to engage in tasks, because this phasic input that kind of contrasts against the tonic isn't really having an effect because there's just a too high a level of tonic firing. And so then, yeah, I think some of the literature in terms of different drugs that are used with ADHD, and I'm struggling to remember the precise names, but some of them have shown to improve symptoms and also reduce tonic firing in the locus coeruleus, which is why there is this, maybe this idea has emerged. But yeah, I mean, I've looked into it a bit recently. I'd say like the literature base isn't that strong, but it's hopefully gonna start to improve. Because yeah, it definitely makes sense when you think about this explore versus exploit. But yeah, I sometimes wonder, yeah, is that the driver? Is it the cause? You know what I mean? Like it could be the case that the tonic is firing because of something else, and then that's what's putting them in this explore mode. It might not be just because of the locus coeruleus. So that's the thing that's difficult to parse out.
Yeah. So it is interesting, though, just even this idea about the subtle differences in neurocognitive systems between arousal and attention, because you could see how those could get conflated quite easily, but they are understandably different, right? That whole the idea about the arousal being on that spectrum of hypervigilant to comatose, and then the attention being perhaps more, as you say, somewhat related, but in some ways not so related, right? Or as a subset. Yeah. And yeah, so figuring that out and then mapping that onto different circuits and then understanding the relevance, as you say, in real time in these kinds of studies with those types of tonic versus phasic changes in how activity is being measured over time.
Yeah. It's just, yeah, it's an interesting area. We've tried to, in our experiments, try to keep the tonic sort of consistent between the sham and the tVNS. So that's why we typically have more time after our trials to let everything settle down. But maybe, and so when we do the trains, our thought is more that we're more affecting the phasic part.
I see.
But yeah, it's something maybe we'll try to manipulate the tonic in future. I'm not sure. Yeah, I think that there's maybe a couple of people in the lab who are interested in looking at these types of tasks in people with ADHD or with anxiety. There's a similar hypothesis about anxiety as well. And also with PTSD as well, as that's emerged as maybe having some sort of locus coeruleus space or deficits or kind of unusual activity in the locus coeruleus.
Definitely. Yeah. You mentioned Parkinson's disease, which is interesting because typically the focus with Parkinson's is on the dopaminergic system, not so much the noradrenergic system. It's understandably involved because of all the various different neuropsychiatric symptoms that usually arise with Parkinson's. What would be the sense that tVNS might be applicable to help with symptoms of Parkinson's?
Yeah, I think there's a few things. I think one thing more, maybe not as a treatment, but as a way to identify a deficit, because what typically happens in Parkinson's disease is that you have these kind of precursor non-motor symptoms, and then what's happening in the background is the degradation then of the dopaminergic neurons has already started. And then by the time that you typically get you start to see an onset of motor symptoms, that's typically the point where you get a diagnosis and then you get treatment. And yeah, there's treatments for the reasonably effective treatments for the deficit of dopaminergic neurons, which can kind of stave off the worst of the motor symptoms and eventually let them plateau. Whereas if there's no treatment, they just go very bad, very fast. But I think the kind of more what we the locus coeruleus starts to degrade earlier, and actually a lot of the non-motor symptoms match up with what we would think of as changes in noradrenergic activity. So we have often difficulty sleeping, which is, if we think of this curve where the tonic is setting, moving us into more like a sleep state. You have, sometimes cognitive deficits, cognitive difficulty with different kind of cognitive tasks, which is also in some way related or could be related. And then, you have these kind of symptoms, and maybe there could be some sort of system where, and then, as I said, we have this way of measuring the effect or measuring activity in the locus coeruleus through the pupils. So maybe you could have kind of checkups earlier ahead of time where you have a tVNS set up, and then you look at how the tVNS is affecting the pupil size. And then maybe you might have someone who comes in to some sort of routine checkup and, they're not actually showing a response in their pupils to the tVNS. That could be maybe a way to assess if they're starting to see degradation in the locus coeruleus, versus there's not really a way to do that with imaging. And then you can imagine like the time point at which you start getting treatment really affects how the disease progresses. So if you come in and then maybe you can go to another sort of more dopaminergic based test. You're like, okay, they show deficits in the pupil size test, and now we're going to see if they actually have the loss of dopaminergic neurons. And then that would allow you to more quickly diagnose maybe Parkinson's before you start to see the onset of the motor symptoms. And so, obviously that would improve the outcome because people are getting treatment earlier, and that the time at which you start getting treatment really affects how you progress. I see that's not really a treatment, but more like a way to assess a biomarker of whether there should be a diagnosis. And then the other kind of aspect is the treatment for Parkinson's disease is very much centered on the motor symptoms or addressing the dopaminergic deficit, whereas the non-motor symptoms can often just continue to progress and they're not really targeted. So maybe you could also have tVNS later on as a way to treat the non-motor symptoms as well. That's a bit more uncertain, I'd say, but I think, it's worth maybe looking into.
Yeah, absolutely. Thanks for explaining that. That's exciting, both in terms of a functional biomarker for preclinical diagnostic purposes, but also targeting, as you say, these under-recognized non-motor symptoms. So for clarity, it would be things like disorders of arousal, like, as you say, like the sleep problems that are often arise, attentional, more subtle kind of neurocognitive deficits.
Yeah, exactly. So maybe you'd have someone come into their family doctor and they report these symptoms, and I suppose right now probably not much is done, or there's certain things that can be done, but there maybe is not a focus on whether this might be the start of Parkinson's. And in an alternate reality, then even the doctor in their office could have some kind of setup with a pupil camera that measures pupil size and like a small stimulator, just to get a sense of whether there is maybe, this person doesn't show any change in their pupil size in response to the tVNS compared with sham. Actually, I'm going to refer them to go and see if they have the start of the degradation because the degradation is occurring in the dopaminergic neurons. It's just not really noticeable, so you can catch it earlier. So it could be like more of a change in the procedure. I think that's an idea that there is out there, but not yet.
Yeah, no, it's very interesting. It strikes me that the more we understand about this and the more that there are these interesting technologies, that perhaps we're moving towards more precise, both in terms of the diagnostic side, early diagnosis allowing for earlier intervention, but also precision neuromodulation of specific cognitive functions like attention, arousal, these kinds of things. So it's really interesting.
Yeah, I think there's a lot of potential ways to go, like certainly we do more basic neuroscience, trying to figure out how the brain works, but also applying that in the clinical space. I think there's a lot of questions emerging that are very interesting.
Yeah, for sure. Thanks so much, Dr. Denyer. Ronan, thanks again for joining us today here. And it's just been super interesting to hear about your work and your lab's work and future directions. What kinds of projects are you focusing on these days that you're embarking on? Is there anything in particular, any one study in particular that you're particularly looking forward to starting or getting further into?
Yeah, so I'm just in the middle of collecting data for a new project, and it's a behavioral project with tVNS. So yeah, we're using this paradigm called a force response task, and actually it's a reaching variant. So we have this device called a kin arm where you can have people reach into this space and measure the kinematics of their reach. And so but they basically hear these four tones and they're instructed you always need to move in some direction on the fourth tone. And then shortly before that fourth tone, we show the target that they should go to if they can. And so we vary the time that we show the target. And so you can imagine if you have half a second, then you're going to hit the target. Whereas if you have maybe only 100 milliseconds, it's going to be hard to hit the target. And so by basically analyzing their success rate at these different times, we can create like a speed accuracy curve. And so one thing we're looking at is whether tVNS shifts that speed accuracy curve. So if you give a person 100 milliseconds with tVNS versus sham, does that increase their success rate? And then a related thing within the task, we're also looking at the effect on the vigor of the reach. So how fast the person reaches or how the peak velocity of the reach. And so yeah, there's this kind of idea that when we increase the speed at which we decide, we also increase the speed at which we move. And this we call like a co-regulation of decision and movement vigor. And so, yeah, I'll be looking into the reach too and whether that is increased with the tVNS. So the, yeah, more basic motor neuroscience study, but it's a really cool task. And I think having the reach, it gives like a real kind of richness to the data, which is new for me. Yeah, I'm looking forward to exploring the data more.
Yeah, that's super interesting. And you can see how learning more about the basic neuroscience in that regard could have potential clinical applications for things like stroke rehabilitation that you were discussing beforehand.
Yeah. So in Dr. Boyd's lab in UBC, she has a kin arm, and we use it all the time for different studies in stroke. And so it can be used as a device to diagnose, but also to administer rehab on. So yeah, it's certainly relevant to that.
Yeah, that's great. Thanks again, Dr. Denyer, Ronan. It's been really interesting. Today's conversation really highlighted something profound. I think that relatively simple, non-invasive interventions, not simple, but I guess seemingly simple, non-invasive interventions like tVNS really can give us causal access to understanding deep brain systems like the locus coeruleus. And I think for me, what's particularly compelling is really the shift in understanding that it's not just speeding up cognition, but more like optimizing it, improving the signal to noise, stabilizing the attention, helping us understand the subtle differences between systems like arousal and attention, and maybe even helping the brain recover from errors in real time, which I think has really enormous implications, not just for neuroscience, but for how we think about treating conditions like ADHD, depression, stroke, neurocognitive disorders that we've been discussing, and maybe even enhancing performance in healthy individuals. That's a whole other discussion as well around ethics and such. But anyway, maybe we'll have to have a follow-up discussion about those other interesting things, Ronan. And yeah, but in the meantime, just want to wish you all the best. And thank you so much for joining me today. It's been such an interesting conversation.
Yeah, thanks so much for having me. It was a great time today.
Okay, take care, all the best, and good luck with all the projects that you and your lab are involved with. And yeah, we're just looking forward to seeing the papers that come out of your research.
Yeah, thanks so much. Yeah, look forward to it.
Okay, take care, all the best. Bye-bye now.
Bye.
Happy St. Patrick's Day, by the way.
Yeah, pick the perfect day. Yeah.
Yeah, perfect. Awesome. Okay, cheers. All right.
All right, see you later.
Take care. All right, bye-bye. Thanks.
Bye.